talk a little bit about these two core methods programs, and these are the ones that our audience is probably most familiar with. Starting with the AOAC Performance Tested Methods Program, or PTM. At one time, you may have heard this referred to as the AOAC RI approval, and because it's administered by the AOAC Research Institute, it's sort of begotten that name. However, Performance Tested Method status is the single laboratory validation with an independent laboratory study. That's sort of the primary method test kit validation that you'll see for many of the food safety test kits, whether they be for mycotoxins, allergens, ATP sanitation systems, pathogenic bacteria, or general microbiology tend to be the major areas. You'll also see them for antibiotic residues and different drug residues and things of that nature. In addition to that, there's the AOAC Official Methods of Analysis. Some of you may have heard or seen this commonly referred to as OMA. And this is also a method approval and publication validation program. This involves the use of evaluating standard methods performance requirements and then making sure that the individual method is validated against those requirements. These typically are involved in collaborative multi-laboratory studies to give us additional statistical measures that laboratory to laboratory performance is comparable. These are always published in Journal of AOAC, as well as the official methods of analysis that you'll see regularly published through AOAC. And they are generally thought of as not only test kits, which you tend to see predominantly in the PTM program, but any method, whether it be a LCMS methodology or some other type of technology that could be used for laboratory validation. I've seen different proteomic and genomic assays go through this. I have seen a variety of instrumentation and instrument-based methods go through this. Even if you go through the official methods of analysis, things going back to thin layer chromatography and different titration methodologies can go back a number of years. And so these tend to not necessarily be proprietary methods in the form of test kits, although they can also be very open methods that anyone can simply read the publication and then execute at their laboratory.